Good morning everyone, I'm Gina Thornycroft and I'm going to talk to you a little bit about the two-spotted bug, Bathycoelia distincta, as a pest of major economic importance in macadamia. Some common macadamia pests are listed here: macadamia nut borer, false codling moth, thrips, mealybug, green vegetable bug, yellow-edged stink bug, and most importantly the two-spotted bug, B. distincta. A little background on macadamia production in Malawi. We are the 7th largest producer worldwide, the 3rd largest in Africa. This accounts for 3% of the global market share and represents a market value of $30 million in Malawi. Insect pests present a major challenge in production, particularly the two-spotted bug. This figure varies between producers and factories, but for the sake of the example I'm about to give you, we'll say we worked out that 50% of the unsound kernel passing through the factory was due to stink bug damage, while other defects such as mold and the like remained pretty constant year on year. You'll see here a couple of examples of what stink bug damage looks on a kernel. You can see these classic white spongy lesions which are characteristic of stink bug damage. This is what happens when tissue is liquefied and the oil is extracted by stink bugs. I want to give you a couple of examples of a good versus a bad year. These are the results from what we'll call Division A for confidentiality. Now, some orchards in this division were thinned between the 2 years, which will account for a slight reduction in yield. As you can see, in 2014, 410,000 kilos of nut and shell was recovered versus 359,000 kilos in 2015. Of that nut and shell, 147,000 kilos of kernel was recovered in 2014 versus 127 in 2015. This gives a total crackout of 35.9% and 35.4% respectively. This is a very good result. Now, based on these figures, you would think that the 2 years are fairly similar. However, unsound kernel makes all the difference. In 2014, of the 410,000 kilos of nut and shell recovered, 88,000 kilos was achieved in terms of sound kernel. Unsound kernel, 59,000 kilos, which gives us 21.5% sound kernel and 14.4% unsound kernel. Not great. In 2015, of the 359,000 kilos of nut and shell recovered, 117,000 kilos was sound, 10,000 kilos was unsound. This gives us 32.6% sound kernel recovery and 2.8% unsound kernel. This is brilliant. Some changes made between 2014 and 15. The orchards were thinned, as I mentioned, which gave a slight reduction in yield. However, the open canopies allowed for better chemical application. The estate sprayed up to and during the harvest, which they didn't in 2015, which accounted for a lot of the late season damage. Spray rounds were applied closer together. And between these 2 years, we noted that mold and all other defects running through the factory did not change significantly. The difference in unsound kernel was a direct result of bug damage. Here are some pie charts just to illustrate that. You can see the pie chart on the left represents the nut and shell and the kernel recovered from that nut and shell volume. The pie chart on the right shows you the split of sound versus unsound kernel. Now you can see in 2014 there is a sizable chunk of unsound kernel. 2015, that chunk is significantly smaller. Now 2.8% is a fabulous result, but we are not going to expect that every year. However, 5% is a good value to aim for. So let's say if 5% had been achieved in 2014, that would have given us an extra 38,500 kilos difference in saleable kernel. Now this is premium grades. This is not including industrial grades for those that also produce that. This is just the whole, the premium grades. Now, at $8 a kilo, which is a conservative figure, this would account for $308,000 loss of profit in 2014. Now, that's just for one division, not across the entire estate. Aside from the loss in profit, there are knock-on effects of excessive stink bug damage. The factory processing time is significantly increased. Hygiene, the more you've got to handle, the more you've got to sort your nut, the greater the risk of contamination. There's also the failure to meet targets in terms of yield. These losses, as you know, are unpredicted because it's impossible to see stink bug damage until you have processed the nut. Current strategies employed to control two-spotted bug include chemical pesticides. Now, there are limited chemical groups available that are effective against the two-spotted bug. It's notoriously difficult to control. Primarily, organophosphates and pyrethroids are utilized. Unfortunately, these are often repeated, and pesticides with the same mode of action tend to lead to insect resistance. Dilution rates should be calculated on an individual basis and should take into account tree size, canopy density, orchard tree spacing in order to make your spray rounds effective. If you're over-applying your chemistry, obviously that's going to be a lot of money that you're wasting. If you are under-applying, you could be applying sublethal doses. Sublethal doses of pyrethroids in particular have been linked to an increase in the rate of two-spotted bug development, resulting in more rapid life cycles and making it that much harder to control. Scouting is generally done on the majority of estates. Scouts monitor for pests in the field, either manually or using knockdown sprays. Currently, a lot of this information is collected and it's really valuable. However, it's not necessarily utilized to its full potential. So what do we know about two-spotted bug? If, as an estate manager, you do not know a lot about the two-spotted bug, then how do you plan your spraying? Chemical selection and spray timing are heavily reliant on having a really good understanding of your target pest in order to be effective. Calendar-based sprays, which are often designed for the South African market, are not necessarily appropriate in Malawi. We have different climatic conditions. Our temperatures tend to be higher. We lack the cold winter temperatures, which help to break the life cycle of the two-spotted bug. Our orchards are often much larger as well. The trees are larger. The estates are larger, and this makes it all really, really difficult to manage up here. So let's start with some two-spotted bug biology. We collected live masses in the field and observed what happened when they hatched. The nymphs could be categorized into 5 stages of development known as instars and became adults after an average of 33 days. Now, this is highly temperature dependent, but this is the average that we observed under ambient conditions at the start of the experiment. You can see the 5 nymphal stages here, which are not winged. The adult is winged and very mobile. So ideally, you want to be controlling the bug at the nymphal stage. The adult here, you can see it has 2 spots on its scutellum on the top. This is fairly similar to a yellow-edged stink bug, except that if you look closely, you'll see that there is a black spot within a white spot, and that is where the name two-spot comes from. Once the first adults had emerged after 33 days, we observed them to note any differences between male and females. Females tend to be larger and have a more pointed abdomen shape, whereas the males are slightly smaller and have a concave abdomen. They mated and produced eggs in the lab. We noticed that the females under lab conditions were able to lay eggs as often as every 2 days. These are laid in clusters of 14 eggs, and it's fairly consistent. The adult lifespan we noted was roughly 30 days in the lab, but this is likely to vary quite significantly in the field. The eggs are about 1 by 1.5 millimeter ovals with a crown of small white projections around the top of the egg. These are called micropillar processes. They took an average of 6 to 7 days to hatch at ambient temperatures, and dark eye spots are visible in live eggs when they are nearing hatch. Hatching is done by a buildup of fluid which generates enough pressure to crack the egg along the line of micropillar processes, resulting in a neat uniform flap. This is characteristic of stink bug eggs which have hatched naturally. If you are collecting live masses that have more jagged edges, they don't have this neat flap, it's quite likely that they have been parasitized and that parasitoid has consumed the stink bug nymph inside the egg and chewed its way out. So this is a sign that you might have parasitoids living in the orchard, which is great. The nymphs darken gradually after hatching. These photos are taken every 30 minutes to illustrate that. These nymphs do not have wings, as I mentioned. They don't move from the egg mass and they do not feed on macadamia at this stage. This is the ideal stage that you want to be controlling these bugs before they become mobile and before they are destructive. Nymphs molt to allow for growth between the instar stages. Each nymph will molt 5 times. Molting is known as ecdysis, and the skins which are shed are called exuvia. Now, the observations that we made were used to create some handbooks and posters. These we found would promote good identification skills in the field and also encourage an interest in P&D for the scouts. They're your eyes on the ground, and it's really, really good to try and encourage that kind of interest and to show that this is really important in the management of two-spotted bug. Now, the most significant observations we made were the stage at which the bug is able to feed, the feeding method itself, the rate of development, and natural enemies and biological control which might be used to help control the bug. The feeding stage. First instar nymphs, as I mentioned, remain on egg clusters and do not feed until after their first molt. At this point, we provided them with a macadamia nut, and these second instar nymphs appear to be feeding. But how do we prove that they are feeding on the macadamia kernel and not just absorbing nutrients from the husk? Firstly, we assessed this nut for damage, and secondly, we presented a nut and shell with the husk removed to eliminate the possibility that they were not actually feeding on the kernel. This slide shows a picture of the nut taken from the previous slide. This is the exact same nut that was exposed only to second instar nymphs. Now you can see the characteristic white spongy lesions around the edge of the nut, which show that it has been damaged by stink bugs. The nut and shell that we placed in the tank also attracted nymphs and also showed similar signs of damage once we cracked it. So how are they doing this? At 5 days old and only 4 millimeters in body length, with a rostrum length of a maximum of 4 to 5 millimeters. How are they getting through the husk, the shell, and into the kernel? This brings us to the feeding method. The stink bug mouthparts is known as a rostrum and is comprised of 3 parts: the labrum, which can be thought of as a top lip, a stylet, which is basically modified teeth, and a labium, which is basically the bottom lip. Now, labium, as you can see from these pictures, does not appear to be used to penetrate the shell. In the first picture, you can see the stylet, dark hair-like structure, and as the stylet pierces the husk, the labium appears to be used as sort of like a scaffolding, and as it progresses through the husk and through the shell, the labium is removed to allow the bug to lie flat against the husk and for the stylet to penetrate right to the center of the kernel. Now, based on these images, you can see that stylet length is the only limiting factor in when these bugs are able to feed, not rostrum or labium length. If we look back at the photo of the second instar nymphs feeding, you can clearly see a large loop where that small white arrow is visible when they begin to feed. Now, this indicates that the stylet is indeed significantly longer than the labium. Here's another photo to illustrate that point. You can see the stylet being cleaned by the second instar nymph. It forms a loop and then goes over the back of the bug, and you can see the tip indicated by the white arrow. Only the stylet penetrates the husk and shell during feeding. The stylet is made up of 4 filaments, which can be thought of as specially modified teeth. These interlock to form the stylet bundle. Now, the serrated mandibular stylets pierce the surface, advancing one at a time, while the maxillary stylets move into the gap as they progress through the husk and through the shell. This is a cross-section of the stylet bundle, which clearly shows the 4 filaments and how they interlock. This is stylized, but gives you the idea that there are 2 channels which form. The bug is able to inject a digestive enzyme via the salivary channel. This liquefies the tissue in the kernel and is then extracted via the feeding channel. The next thing we looked at was the rate of development for two-spotted bug. The rate of insect development is linked to temperature, not time. Each insect requires a specific amount of accumulated heat to complete its life cycle. Degree days are the units used to describe this accumulated heat. Each insect also has a specific lower developmental threshold below which no development takes place. Once this LDT is known, it's possible to calculate the rate of insect development using the daily average temperature. Now, if we take the conspurse stink bug in California as an example, because it has a known lower developmental threshold of 12 degrees, say the average temperature today is 13 degrees, 1 degree above the LDT. This means 1 degree day will accumulate in this 24-hour period. If the daily temperature was 22 degrees, 10 degree days will accumulate in this same 24-hour period. Now this just illustrates how just how much the temperature can influence the rate of development of an insect. Now I've used the conspurse stink bug as an example because we don't know the lower developmental threshold for two-spotted bug, particularly in Malawi. We've started the work on it, but we need to confirm it. Now we conducted an experiment in Cholo to identify the lower developmental threshold for two-spot in Malawi, or we began it. Nymphs were placed in tanks and maintained at different temperatures using thermostats and heat lamps. It's a very simple design, but we found it worked really well. The slowest rate of development observed was 72 days, and the fastest rate we observed was 28 days. Now this just serves to highlight how drastically the rate of development can change when you have a slight difference in temperature. If we are to complete a degree day model for Malawi, this would provide really valuable insight into the rate of development and how this changes based on the climatic conditions. This would allow us to be more precise in the timing of our sprays, and I think it would be a really valuable tool in the management of two-spotted bug. One of the natural predators of stink bug is the Trisulcus wasp. These were recovered in an orchard and appear to be quite widespread throughout Malawi. We've heard of them being recovered in orchards all over the country. Parasitized eggs appear to be darker and have a characteristic dark band across the center, as you can see in the bottom center photo. These eggs were collected and hatched. We observed the adults as they mated and laid new eggs inside stink bug eggs. The nymphs develop inside the egg and eat the stink bug nymph before chewing their way out of the egg. These showed really good potential as a possible biocontrol agent for stink bug. We've looked into bringing in, this type of wasp in from somewhere like South Africa, for example. There are products which exist. You can buy these in mass and rear them and release them in your orchards. However, this is not registered in Malawi yet, so there may be potential to begin a mass rearing project within Malawi. This is something that I think is worth looking into. Another potential biological control agent, Bavaria bassiana. This is an entomopathogenic fungus which kills insects. This is highly specific, so it's not harmful to beneficials, which is great. There are a few challenges, logistics and storage. There are several products that contain Bavaria bassiana. Some of them require refrigeration, some of them require multiple spray rounds to be effective. This is all something that needs to be trialed quite extensively, and potentially could work quite well in conjunction with the degree day model, used as a way to inform when you should apply this fungus. Now, just an overview of some of the challenges we've looked at. Chemicals are being removed from the market all the time. The repeated use of organophosphates and pyrethroids is contributing to resistance and just making the problem worse. There is increasing pressure for good quality products using green farming practices. Obviously, the pesticides that we're applying are disruptive to beneficial insects, and there is a clear need for industry-wide research in order to better our management of this pest. I'm going to finish with 2 slides just to give you an overview of some of the best practices we've observed as we've traveled around estates throughout Malawi. Chemicals. A better understanding of chemical mode of action really helps in informing a spray program. There are resources such as this IRAC chart you can see on the page. This is the Insecticide Resistance Action Committee. You can use this as a tool to help you see which chemicals have different modes of action, how to judge whether they're effective or not. Not all of them will cause an immediate knockdown effect, and it's important to understand that to know whether they're really being effective. Knowing your dilution rates and ensuring your spray equipment is in good condition, making sure your water volumes are correct, your application rates are correct. This will all help you to maximize the efficacy of the product you're applying. Scouts are great. They work really hard. In general, the teams that we've seen have been enthusiastic. There is always room for improvement. We can look into pest identification skills and the importance in recording things accurately. We can also look at the digitization of data. There is a huge amount of data at a lot of estates that goes back for many, many years. This is a great resource. We can analyze this to view and predict trends in pest populations. We can also integrate this with GPS data in order to pinpoint hotspots and apply spot treatments before they get out of hand and avoid the application of a blanket spray. Irrigation is great where it's feasible, where it's needed. Areas more prone to drought can be supplemented with irrigation to reduce nut drop and maintain healthy trees. In general, healthy, less stressed trees are going to be less attractive to pests, as you would imagine. Completing the degree day model will really help utilize climatic data to predict outbreaks and help us to control the pest before it gets out of control. Biological control, we've looked at the Trisolcus wasp as well as Bavaria bassiana as potential biological control agents. They both show good potential and are definitely worth pursuing. Varietal selection, this is really important. Selecting appropriate varieties for your location and climate to make sure that your trees remain healthy. Dwarf trees, these are easier to manage. They're always going to be easier to manage. They're smaller, you can get a better spray coverage, they're easier to monitor and easier to harvest. Orchard management in general, pruning and selective limb removal. Maintaining an open canopy is really important. It allows you to get a better spray coverage when applying a pesticide or a biological product. It increases photosynthesis and the amount of bearing wood, which will in turn increase your yield. I'm sure most of you have noticed that in very large trees, you quite often tend to get the bearing wood on the outside, all the leaves on the outside. As you walk underneath, you'll notice the canopy is hollow and you're losing out on a huge volume of crop that you could otherwise be getting if the canopy was more open and exposed to sunlight. Ground cover. More and more estates are introducing ground cover. It aids in soil retention, it reduces the soil temperature, improves moisture retention and provides valuable host plants and shelter for beneficial insects, which in turn will increase the biodiversity in your orchards, and this ought to help keep your pest populations at an appropriate level.